My name is Yuki Tatenaka from Dojindo Molecular Technologies. I would like to present my work to you, titled Development of Fluorescent Glucose Uptake Probes and Quantitative Detection of Various Nutrients Uptake. Let us begin with this figure that illustrates how cells take in various nutrients to use for energy and life-supporting activities. The metabolism of these nutrients varies depending on the extracellular environment, the state of the cells, and the type of cell. Nutrient metabolism not only has a large impact on energy production, but also in the regulation of many cell functions, including gene expression. We at Dojindo have been focusing on the uptake nutrients by cells and have been developing various assay systems. We have developed a new fluorescent probe to detect the cellular uptake of glucose with high sensitivity. We would like to introduce this item and outlines its usefulness. Here I will explain the relationship between diabetes and glucose uptake. Typically, diabetes is caused by decreased insulin secretion and decreased responsiveness to insulin. When insulin binds to insulin receptors on the cell membrane of muscle cells or adipocyte, a signal is transduced that results in glucose transporter 4, GLUT4, being translocated from vesicles to the cell membrane. At the cell membrane, GLUT4 enhances the cellular uptake of glucose. In the case of diabetes, however, cellular uptake of glucose is inhibited, and this has consequences of glucose metabolism and regulation. In order to research diabetes, the ability to evaluate insulin resistance and the ability to screen for drugs that affect glucose uptake are both pivotal. Currently, there are three main methods to measure glucose uptake. The first method uses radioisotope labeled glucose. The main feature of this method is glucose uptake can be quantified. Also, this method is used by many. The radioactive material requires special handling facilities and disposal services, making the method inconvenient and costly. The second method is metabolome analysis using LC-MS. This method can measure a variety of metabolites, including glucose and amino acids, in a single experiment. The problem, however, is that the results are difficult and expensive to analyze. The third and most recent method involves use of 2-NVDG, a fluorescently labeled glucose analog. This method, however, has low sensitivity due to the low fluorescence intensity of 2-NVDG. We at Dojindo are excited to share that we have developed our own novel fluorescent probes with high fluorescently intensity, allowing us to visualize glucose uptake with high sensitivity. Dojindo's glucose uptake probes were developed to resolve the issue of cost, convenience, and sensitivity all at once. We have developed three colors of fluorescent probes to visualize glucose uptake with glucose uptake probe blue, green, and red. These probes are taken up into cells via glucose transporters, and their intense fluorescence can be detected by fluorescence imaging, flow cytometry, or microplate assay. Here we see the fluorescence properties of three probes. The spectra of three probes each fit to common filter set in fluorescence microscopy. The probes can be used for multiple staining with other biomarkers, and the different color allow for different combinations. The operation is very simple, requiring only three steps: pretreatment, staining, incorporation, and washing. It even manages to save significant time over to NVDG despite having the same process. The cells incubated for 15 minutes in serum-free and glucose-free medium. They were then incubated for another 15 minutes in serum-free medium that was either glucose-free or contained 25 millimolar glucose. After that, the cell was washed with our washing and imaging solution, which we called WI solution, at 4 degrees in order to prevent the probe from leaking out of the cells. When the probe was added under the glucose-free condition, a clear fluorescence signal was obtained from within the cells, confirming that the probe was incorporated into the cells. When the probe was added in the presence of glucose, however, the probe delivered fluorescence signal was reduced. This indicated that glucose competition inhibits the uptake of the probe, meaning that the probe was being taken into the cell via the same mechanism as glucose. The measurements were obtained via plate reader, and the data correlate with the imaging results. The strong correlation verifies that measurements can be performed using a plate reader. Additionally, it was confirmed that glucose uptake probe green can measure glucose uptake with higher sensitivity than existing fluorescence reagent to NVDG. These are the results of glucose uptake experiment where insulin was added to adipocyte differentiated from 3T3L1 cells. After adding insulin, we observe glucose uptake using our fluorescent probes. Fluorescent signals were increased in the insulin-treated cells, indicated that glucose uptake was enhanced. This is believed to be because the fluorescent is taken up through GLUT4 glucose transporters, which are translocated to the cell membrane surface due to insulin. Here are the results obtained from using cytochastrin B, a glucose transporter inhibitor on HepG2 cells. After incubating the cells in serum-free media containing 5 micromolar cytochastrin B for 24 hours, we observed the glucose uptake ability using the 3-colored versions of our probe. Compared to the controls, the cells treated with cytochastrin B had a reduced fluorescent signal. The degrees indicated that the uptake of the probe was inhibited, further suggesting that the probes are taken up into the cell via glucose transporter. According to some research, people who are obese are up to 80 more likely to develop type 2 diabetes. Thus, it should be important to also observe lipid stimulation and glucose uptake in diabetes research. Knowing this, we tried to detect lipid droplets and glucose uptake simultaneously in a single sample. These images are the results of co-staining with glucose uptake probes and lipid droplet probes in a differentiated T3L1 adipocytes. Given the various color versions, Dojindo glucose uptake probes can easily co-stain with green probes, red probes, or any other log wavelength fluorescent probe. As a result, we were able to simultaneously measure glucose uptake and detect lipid droplets. In addition, we have also developed safe, simple, and reliable methods to measure the uptake of cysteine or amino acids, which are nutrients just as important as glucose. I would like to introduce you to these methods, the amino acid uptake assay and the cysteine uptake assay. This current slide covers the new amino acid uptake assay. L-type amino acid transporter 1, LAT1, is one of the amino acid transporters, and LAT1 is specifically expressed in many cancer cells. The conventional method for assessing amino acid transporter activity involves the use of radioisotope labeled amino acids. However, this method requires special handling and the disposal of radioactive materials. To avoid these limitations, we developed a simple fluorescence assay for amino acid transporter activity using a paraboron phenylalanine (PPA), which is an amino acid analog used in neutron capture cancer therapy, as well as fluorescent probe for PPA. Our method can be applied to fluorescent imaging, microplate measurement, and flow cytometry. It is thus useful both evaluating cellular amino acid uptake activities and screening amino acid transporter inhibitors. This slide shows the principle of measuring cellular cysteine uptake. The cysteine transporter XCT is a crucial transporter that maintains cellular redox balance. It does this by regulating cysteine uptake and thus regulating glutathione synthesis. We have developed a new and convenient fluorescent method for measuring XCT activity using a cysteine analog, cysteine. Cysteine is known to be transported into cell through XCT-like cystinders, and cysteine can be specifically detected by using a fluorescent probe. This method, which is based on microplate assay, can be used to measure XCT activity in multiple cells as well as the screening of potential anti-cancer drugs. We believe these novel technologies can be utilized in drug screening for making significant advances in biomedical studies and the diagnosis of various diseases, especially diabetes. Let me summarize the main points of today's presentation. We believe these novel technologies can be utilized in drug screening for biomedical studies as well as in diagnosis of various diseases, especially diabetes. I'd like to acknowledge Dr. Hirakawa, Dr. Shimomura, and Dr. Ouchi for their work. If you are interested in our new methods or have any questions, please feel free to contact me. Thank you very much for listening.